Hello, everyone, and welcome to this webinar hosted by Jen, Genetic Engineering and Biotechnology News. Our topic today is emerging modalities in drug discovery. Today's webinar is sponsored by WuXi AppTec. Thank you very much for joining us. I'm delighted to be the host of today's webinar. I'm Kevin Davis, the editorial director for Jen and founding executive editor of the CRISPR Journal and Gen Biotechnology, Gen's new marquee peer-review journal, publishing original research in all of the aspects of biotechnology that you've come to know and value Gen for its coverage over the past 4 decades. I'm also the author of the book Editing Humanity, The CRISPR Revolution and the New Era of Genome Editing, which is out in paperback. We have a very distinguished guest speaker today, Dr. Tao Guo, who is with WuXi AppTec. In today's webinar, we're going to be hearing about some new and emerging modalities in drug discovery, including targeted protein degradation, targeted covalent inhibitors, and nucleic acid therapeutics. Our guest speaker will be discussing solutions for addressing the complexities in designing, synthesizing, and optimizing these molecules, with an added focus on the special modifications and delivery systems that are needed for siRNAs, short interfering RNAs, and allele-specific oligonucleotides, or ASOs. Dr. Tao Guo, our presenter, is the Senior Vice President and Head of the International Discovery Service Unit at WuXi Chemistry, part of WuXi AppTec, based in Shanghai in China. Dr. Guo has more than 30 years cumulative experience in drug discovery, trained as an organic chemist, receiving his PhD from Columbia University and completing a postdoctoral fellowship at UC Berkeley. He's an inventor of some 10 clinical candidate compounds, with one that has been approved by the FDA. For this webinar to be a success, we really want your questions. So please use the time during the presentation to think about questions for our presenter. Enter them at any time during the next 30 minutes or so, and we will take as many questions as we can in the live Q&A session at the end of the presentation. So thank you in advance. But now it is time to welcome our guest speaker. So Dr. Tao Guo from WuXi AppTec, we're really looking forward to your presentation. Over to you.
Hello, everyone. My name is Tao Guo. I'm a Senior VP and Head of WuXi Chemistry, RCS, BD, and IPM. Today, it is my great pleasure to share with you on enabling and accelerating innovative and emerging modalities drug discovery through a global open platform. A quick note about us. WuXi AppTec is a global open platform company with operations across Asia, Europe, and North America. Using our unique CRDMO and CTDMO business models, we provide integrated end-to-end services and solutions to pharma and biotech customers around the world to advance the discoveries and deliver groundbreaking treatments to patients. Our vision is every drug can be made and every disease can be treated. Today, I will talk about 3 emerging modalities: targeted protein degraders, targeted covalent inhibitors, and nucleic acid therapeutics. First, targeted protein degraders. Targeted protein degradation is an emerging therapeutic modality. It has the potential to target undruggable proteins. A major class of TPDs are proteolysis targeting chimera PROTAC protein degraders. These are heterobifunctional small molecules consisting of 2 ligands joined by a linker. One ligand recruits and binds to a protein of interest, POI, while the other recruits and binds an E3 ubiquitin ligase. Simultaneous binding of the POI and the ligase by the PROTAC induces ubiquitination of the POI and its subsequent degradation by the ubiquitin proteasome system, UPS, after which the PROTAC is recycled to target another copy of the POI. This catalytic type of mechanism of action and event-driven pharmacology distinguish PROTACs from classical inhibitors, which have a one-to-one relationship with the POI and whose pharmacology is driven by stoichiometry and usually by interactions with a catalytic site. Another important class of targeted protein degraders are molecular glues. They are based on the serendipitous discovery that thalidomide and its analogs act as degrader molecules. Although not heterobifunctional in the manner of PROTAC molecules, molecular glues promote ubiquitination of POI by enhancing a protein-protein interaction between a ligase and a potential substrate. Since the first small molecule PROTAC was reported in the literature by Cruz and colleagues in 2001, the technology has moved from academia to industry. A number of biotech and pharmaceutical companies have disclosed programs in preclinical and clinical development. In 2019, the first PROTAC molecules, ARF-110 and ARF-477, entered clinical testing. In 2020, these trials provided the first clinical proof of concept for the modality against two well-established cancer targets, the estrogen receptor and the androgen receptor. With this proof of concept success, the TPD field is now poised to tackle undrugged targets and other classes of difficult protein targets. As of now, over 20 targeted protein degraders have advanced into clinical development. Beyond the current slate of compounds in clinical testing and others in the development pipelines, what is the outlook for the next 20 years of TPD? Based on Cruz and colleagues, the next milestones in TPD will be focused on 4 clinical translation inflection points. First, defining and clinically demonstrating that target classes best served by degradation over inhibition. Second, expanding the scope of E3 ubiquitin ligases employed clinically in a targeted fashion to enable precision medicine. Third, extending the clinical reach of modality beyond oncology. And fourth, validating TPD modalities beyond IMiDs and PROTACs in clinical settings. Inspired by PROTACs, new degraders are emerging that harness endocytosis and autophagy to send a wide array of targets to the lysosome for destruction. As PROTACs can't degrade many categories of disease target, so academic and industry scientists are extending the degrader concept to a diverse set of new targets by exploiting the cell's other main recycling center, the lysosome. The new generation of degraders avoids the ubiquitin proteasome system entirely and instead harness cellular pathways for delivering cargo to the lysosome for degradation. For example, LITACs, or lysosome targeting chimeras, link an extracellular domain of a target molecule to a cell surface receptor that shuttles it by endocytosis to lysosomes for degradation. The field of lysosomal degraders is very young, but is quickly growing. Besides LITACs and the other extracellular protein degraders, at least 5 different autophagy-based degraders are in development. Genentech's PROTACs, or proteolysis targeting antibodies, and IP Biologics AppTecs, or antibody-based PROTACs, hijack cell membrane ubiquitin ligases that also direct targets to lysosomes. IP biologics contacts degrade extracellular and membrane proteins by tethering them to cytokine receptors for cell internalization. Several other degraders exploit natural autophagy pathways in which specialized vacuoles engulf protein aggregates and complexes or damaged organelles for lysosomal delivery. Heterobifunctional molecules are moving beyond degraders. Drugs then can stabilize, phosphorylate, and inhibit their targets are emerging from academia and industry. Deubiquitination can spare vulnerable proteins from the trash disposal system. Phosphorylizers and dephosphorylizers can control the activity levels of their targets. And the proximity-based inhibitors can take on targets that otherwise offer toxicity overhangs. The list of heterobifunctional acronyms of chemical inducers of proximity beyond degradation are rapidly growing. PROTACs composed of traditional small molecule ligands are poised to become an important modality with advantages over small molecule inhibitors, especially with the potential to target undruggable proteins. PROTACs have also inspired the exploration of other classes of heterobifunctional molecules that harness non-UPS machinery to degrade POIs. Despite 20 years of research, there remain many challenges for targeted protein degrader discovery. First, it is hard to design. Compound design remains largely empirical, as target binding affinity does not predict activity. Many compounds need to be synthesized and tested to find functionally active degraders. Second, it is hard to make. Synthesis is lengthy, requiring the right sequential assembly of the target protein binder, linker, and E3 ligase binder. Purification requires special solvent and column conditions by trial and error, as the compounds have poor solubility and prone to epimerization. Third, it is hard to optimize. Compounds are typically beyond the rule of 5 with low solubility. Many compounds need to be synthesized and tested in animal studies to find orally bioavailable degraders. To support customers in targeted protein degrader discovery, we have built a robust TPD discovery chemistry platform over the past 8 years. As of today, we have over 1,200 FTEs with strong synthetic experiences in linkers, E3 ligase ligands, and target protein ligands. We have made over 1,000 diverse linkers, 100 E3 ligase ligands, and 60 targeted protein ligands. We have delivered over 110,000 targeted protein degrader compounds. Our analytical and purification team has developed over 200 special methods for analysis and purification of the degrader compounds. Using our TPD platform, we have supported over 100 biotech and pharma customers to successfully discover and advance their targeted protein degraders. Now, let's look at targeted covalent inhibitors. Covalent drugs have been used to treat diseases for more than a century, but tools that facilitate the rational design of covalent drugs have emerged more recently. The purposeful addition of reactive functional groups to existing ligands can enable potent and selective inhibition of target proteins. It has been demonstrated by the covalent epidermal growth factor receptor and Bruton's tyrosine kinase inhibitors. used to treat various cancers. Moreover, the identification of covalent ligands through electrophile first approaches has also led to the discovery of covalent drugs, such as covalent inhibitor for KRAS G12C and the SARS-CoV-2 main protease. Covalent drugs have advantages over reversible drugs. These include increased potency, improved selectivity, prolonged pharmacodynamics, built-in biomarker, novel intellectual property, and the targeting of traditionally undruggable proteins. Most pharmaceutical small molecule research focus on the discovery and optimization of reversible drugs. Until recently, covalent drugs have been an underexplored area due to the perceived toxicity concerns. Of the 1673 small molecule drugs approved by the FDA as of 2022, 117 of them are covalent drugs. These include antibiotics, proton pump antiplatelet drugs, HCV protease, proteasome inhibitors, and the more recent targeted covalent inhibitors. The term targeted covalent inhibitors was first used by Singh in 2011 to describe a new class of covalent drugs that were discovered using rational drug design. The approach originated in the early 1990s and it involved converting specific reversible small molecule inhibitors into covalent drugs through the addition of a low reactivity bond-forming warhead, targeting non-catalytic nucleophiles using structure-based design. The approval of non-TCIs over the past 10 years has fueled huge interest in this emerging modality. As an example, let's take a closer look at the progression of EGFR inhibitor structures. EGFR is a key drug target in oncology. The reversible first-generation EGFR inhibitors, gefitinib and erlotinib, were discovered in early 2000s to be effective against tumors harboring somatic activating mutations in EGFR. Covalent second-generation inhibitors were strategically designed with acrylamide microacceptors to react with a cysteine residue, CYS797, in EGFR. These covalent second-generation inhibitors provided prolonged suppression of EGFR signaling. The third generation of EGFR inhibitors selectively target T790M mutant over wild-type EGFR. These compounds maintain the acrylamide group to covalently bind CYS797, but exchange the quinazoline moiety of first-generation and second-generation compounds for a pyrimidine to promote selectivity for mutant EGFR T790M. Higher affinity for T790M over wild-type EGFR not only results in efficacy in cancers with the EGFR gatekeeper mutation, but also contributes to an improved safety profile. The success of covalent EGFR inhibitors has validated the approach of covalently engaging non-catalytic, non-conserved cysteines adjacent to kinase active sites to increase the potency and modulate the pharmacodynamics of initially reversible ligands. Here is a non-exhaustive list of warheads that can be used for TCI design. The warheads are grouped in panels by target residues. The warheads include, for example, acrylamides and propionamides for cysteine, beta-lactams and boric acids for catalytic site serine, vinyl sulfones for lysine, aziridiniums for histidine. Some warheads may be used to target multiple residues. Despite the tremendous progress made over the past 10 years for targeted covalent inhibitor discovery, many challenges still remain. First, it is hard to design. Compound design remains largely empirical as the fine balance between reactivity and stability needs to be achieved. Many compounds need to be synthesized and tested to find the right molecules. Second, it is hard to make. Synthesis is difficult as the compounds are fairly reactive and complex mixtures are often obtained. Purification needs to be performed rapidly using special solvent and column conditions, given the reactivity of the compounds. And third, it is hard to optimize. Compound optimization to improve selectivity of target inhibition over off-target inhibition requires many compounds to be synthesized and tested, often in vivo, to find the candidates. To support customers in targeted covalent inhibitor discovery, we have built a robust TCI discovery chemistry platform over the past 10 years. Today, we have over 950 FTEs with strong experiences in targeted covalent inhibitor synthesis. We have made over 16 types of warheads, including acrylamides, chloroacetamides, 2-chloropropionamides, and vinyl sulfones. We have delivered over 70,000 covalent compounds to customers. Using our TCI platform, we have supported over 70 biotech and pharma customers to successfully discover and advance their targeted covalent inhibitors, including 11 preclinical candidates and 1 FDA-approved drug. Now, let's move on to nucleic acid therapeutics. Nucleic acid therapeutics are emerging rapidly as an important drug modality. These drugs can manipulate gene expression or produce therapeutic proteins, making these drugs suitable for pathologies with established genetic targets, including infectious diseases, cancers, immune diseases, and neurological disorders. Small interfering RNAs, siRNAs, can reduce gene expression via RNA-induced signaling complex, RISC-mediated mRNA degradation. Antisense oligonucleotides, ASOs, can alter isoforms by binding to splice sites. And adenosine deaminase acting RNA, ASOs, ADAR oligonucleotides, can edit RNA. In all three cases, these small RNAs can be designed with site-specific chemical modifications using solid phase synthesis. mRNAs can encode nucleases that mediate RNA or DNA or RNA editing. mRNAs can also be used to replace dysfunctional protein or encode antigens that confer longer-term immunity to a pathogen, such as SARS-CoV-2. A 1978 report pioneered the concept of treating a target RNA as a receptor for antisense oligonucleotide. Its code determines both the affinity and the specificity of oligonucleotide binding through Watson-Crick base pairing. The concept included the possibility of both the single-stranded agents that are now commonly referred to as antisense oligonucleotides, and the double-stranded agents that are known as small interfering RNAs. Antisense technology is now beginning to deliver on its promise to treat diseases by targeting RNA. 10 single-stranded antisense oligonucleotide drugs representing 4 chemical classes, 2 mechanisms of action, and 4 routes of administration have been approved for commercial use. because the rules that determine to which receptor sequence an ASO may bind through Watson-Crick base pairing were understood. True rational design of ASO drugs was theoretically feasible. Thus, ASOs could be designed to interact with excellent specificity for their receptor sequences and to work against undruggable receptors. New medicinal chemistry for ASO had to be established. Medicinal chemistry efforts directed towards introducing good drug properties into oligonucleotides had been made. There have been broad efforts to modify essentially every position in a dinucleotide, except those required for Watson-Crick base pairing. Key goals for these medicinal chemistry efforts have included enhancing the binding of ASOs to cognate sequences in RNA, increasing resistance to degradation by nucleases, improving pharmacokinetic characteristics, reducing pro-inflammatory effects, and optimizing the pharmacological responses following binding to the target RNA. Based on an understanding of nucleic acid structure and biochemistry, modifications of the phosphodiester backbone and the 2' position in the sugar have been major foci. More recently, there has also been a focus on optimizing ASO delivery through the conjugation of targeting ligands and enhancing the therapeutic index of ASOs through site-specific modifications. This year marks the 25th anniversary of the discovery of RNA interference, RNAi in C. elegans, by Fire and Mello in 1998. In 2001, small interfering RNAs, siRNAs, were identified as mediators of RNAi in mammalian cells. siRNA therapeutics enable sequence-specific gene knockdown by eliminating target RNA molecules through complementary base pairing. In contrast to antisense technologies, siRNA relies on a catalytic mechanism. After target mRNA cleavage, siRNA-loaded RNA-induced silencing complex, RISC, is able to dissociate and bind to another mRNA molecule. In consequence, very low siRNA concentrations in the picomolar range are able to induce efficient gene knockdown. siRNA therapeutics have gained great momentum during the last decade, resulting in 5 approved therapeutics targeting the liver for treatment of severe diseases. And the trajectory depicted by the ongoing trials emphasize that even more siRNA therapeutics, also targeting extrahepatic tissues, are likely to be approved in the future. The success of siRNA drugs critically depends on chemical modifications and/or technologies designed to protect siRNA from degradation and to warrant their systematic stability, to facilitate localization to the targeted tissue, and finally, to safeguard successful intracellular delivery. New medicinal chemistry for siRNA had to be developed. This includes, for example, modification of the 2' position in the sugar using 2' deoxy 2' fluoro, 2' O-methyl, or glyconucleic acid. Modification of the phosphodiester backbone using phosphorothioate linkage, and conjugation with GalNAc for liver delivery. Lipid nanoparticles are a key class of drug delivery system that includes nanoparticles approved by the FDA for liver siRNA delivery and for mRNA vaccine delivery. On the basis of the size of the hydrophilic head group, relative to the size of the hydrophobic tail or tails, lipids form distinct structures, including micelles, liposomes, and lipid nanoparticles. FDA-approved lipid nanoparticles contain variations of 4 basic components: cholesterol, a helper lipid, a polyethylene glycol PEG lipid, and a cationic or ionizable lipid. Chemical modifications, all these 4 components are intensely pursued by biotech and pharma companies to improve their properties and to gain novel IP positions. Huge progress has been made over the past 10 years in the discovery of nucleic acid therapeutics. However, significant challenges remain for this new modality of research. First, special modifications are needed. Nucleic acid therapeutics need stabilization, need stabilization chemistry with modifications, including 2'-fluoro, 2'-O-methyl, 2'-O-methoxyethyl, locked nucleic acid, unlocked nucleic acid, glyconucleic acid, phosphorothioate, and PMO. The synthesis of many sequences are needed to identify the right combination. Second, special conjugations are often required. Nucleic acid therapeutics targeting often needs special conjugation synthesis. For example, liver targeting through GalNAc conjugation. Third, special delivery needs to be considered. Nucleic acid therapeutics delivery is very challenging, often requiring lipid nanoparticles assistance. The design, synthesis, and the right mix of lipid nanoparticles are largely empirical, requiring the synthesis and the testing of many lipids. To support customers' drug discovery needs in nucleic acid therapeutics, we have built a strong platform over the past 5 years. Our discovery platform provides end-to-end services from monomer synthesis, oligosynthesis, to conjugate synthesis. Our monomer collection includes bridged nucleic acid, 2' and 5' mono modifications, nuclear base modifications, GalNAc ligand, lipid and spacer amidite, stereo-defined amidite, and PMO monomers. We have made ASO, siRNA, CpG-ODN, and PMO. We have also made a wide range of conjugates, including GalNAc, steroids, peptide, lipid, PEG, and fluorescence tags. We have supported over 100 biotech and pharma customers and successfully delivered over 30,000 nucleic acid discovery compounds and over 2,000 lipid compounds. Thank you very much for attending the webinar. Look forward to supporting your drug discovery needs. Very happy to answer any questions if you have. Thanks.
Well, that was a great presentation. Thank you very much. We will be moving shortly into the Q&A session. I want to just remind everybody that the webinar will be available on demand shortly. So please share the link with your colleagues if you want to watch the webinar back, or share it with colleagues who perhaps were unable at the last moment to attend the webinar. And it is time now to move into our live Q&A session. So this is your last chance pretty much to get your questions in for Dr. Guo, and stay with us. Just give us a couple of moments, please, to transition into the live Q&A session where we will take your questions. Okay, welcome everyone to the live Q&A session with Dr. Tao Guo from WuXi AppTec. That was a tremendous presentation, Dr. Guo. You really gave us I'm sure just a snapshot of the extraordinary science that is going on with your colleagues at your company. We've had a number of questions come in, and you have still time to enter your question into the ask a question box on the screen. We have a good 20 minutes or so for your questions, so please keep them coming. And Dr. Guo is here to answer them. So let's get started. First question, comment. Thank you for this informative presentation. Could you explain a bit more how to select E3 ligase binders for a PROTAC project?
Yeah, thanks, Kevin. Thanks very much for this opportunity to share with the audience, with attendees, our capabilities and also the new modality field and new developments. And also, thanks everyone for attending the webinar. With regard to the first question, and many companies, many customers are starting projects. Often they ask, how do I select the E3 ligase binders? And so to start with, so the customers or the project leaders start with the most successful ones. Majority of the literature compounds are using the CRBN E3 ligase as the target for the oh, sorry, light is off.
That's okay.
I hope the light is okay. Yeah. So so as the the target E3 ligase for the PROTAC molecule design. So that resulted in majority of the success. If you look at the clinical trial compounds, majority are the CRBN based PROTACs. So if you select CRBN as the E3 ligase, your success rates will be high. Of course, more and more other E3 ligase binders, E3 ligase are being used. And the next, prime example is the VHL, that are being used. And also, I think there's at least one compound got into the clinical trial. So, so these two at least, has advanced into the clinical trials. Of course, there are others are, people are experimenting based on, their tissue distribution, based on other properties. So yeah, the short answer is probably CRBN is the choice for your E3 ligase binder.
Okay, let's stay with PROTACs. Can PROTACs, usually with high molecular weight, be applied to CNS areas?
Yeah. So and the answer is yes and no. So the as we discussed, majority of the PROTACs, they are beyond the rule of 5. They have higher molecular weight. They have high lipophilicity, poor solubility, and so in general, the PROTACs are very hard to get PROTACs into the CNS system. However, there are reports of PROTACs targeting CNS, and they're successful literature on this front. So with probably more modification, more optimization, the properties of the PROTAC molecules, despite their larger molecular weight, their property challenges, people can get some of those PROTACs into the CNS. So I do see some successful examples are being reported. So it's possible. It's just a bit more challenging.
Yeah. Next question. When you said that covalent drugs have a built-in biomarker facility, what did you mean?
Oh, okay. So yeah, that's a very good question. So so what I mean is that so you especially when you form this covalent bond, right? So you have this protein labeled, basically quote unquote labeled by the small molecule. So if you analyze the protein, you see it's labeled. People often use ASMS, the affinity selective mass spectrum, so they can detect very clearly your proteins labeled. So when they do the analysis, so that's a quick analysis that your compound is on target. So it's the target protein being labeled by your covalent inhibitor selectively is a good indication that you have a on-target effect, and that can itself be used as biomarker. That's what I mean by the intrinsic biomarker for covalent inhibitors.
I think that probably clears that up. I hope so. What is your solution to improving the solubility while maintaining ligand's affinity and activity?
Okay, so I guess this question is for PROTAC, right? When we mentioned, the solubility. Okay. So, for PROTAC, I mentioned, the molecules are big. You have a lot of, have high molecular weight. So the, the C-log-P is higher. So how do you do, reduce the C-log-P and make it more soluble? So, so the general approach is to introduce more water-soluble, functional groups or units, and more often than not, is to introduce basic, centers that can be protonated in the at the physiological conditions. So you form this, cationic, compound to improve solubility. Another approach is to make the molecule more rigid, because the first generation of PROTAC molecules often use flexible linkers, often use just carbon chain flexible linkers. So those linkers, given the flexibility of those compounds and the carbon hydrocarbon nature of the linker, make them less soluble. As you can see, some of the examples advancing into clinical trials, they have more rigid linkers, and also with more nitrogen atom introduced into the linker portion. Of course, you can also, to some degree, to modify the POI binding part of the molecule, and also to some degree, you can make some modification at the E3 ligase binder part to introduce soluble, solubilizing groups.
Good, thank you. Can you, a quick note, one question asked if slides, the presentation will be available. It won't be available, but just a reminder to everybody watching, the full on-demand version of this webinar will be up in about 24 or 48 hours. So you will have weeks, if not months, to study every aspect, every pixel of Dr. Guo's presentation and share with your colleagues. So plenty of access for all registrants in the days to come. Okay, next question. Can you tell us more about WuXi's capabilities in discovery and development chemistry support in this area, especially for virtual companies?
Yeah, thanks, Kevin. Yeah, so as I mentioned, this webinar is more focused on the discovery. So for the discovery part, we have right now, for the PROTAC, we have some 1,200 chemists with experience in the synthesis of ligands, the POI ligands, the E3 ligase ligands, as well as linkers. Of course, we have experience putting them all together. We have delivered some 110,000 these heterobifunctional type of compounds. So we have strong expertise in the discovery phase. I also mentioned, our analytical colleagues, they have some 200 methods developed specifically for the heterobifunctional molecules. And in addition to that, we have supported a number of customers to move their compound beyond the discovery, beyond the preclinical candidate stage. We have helped a number of companies move compound to preclinical studies, so that require scale up, a non-GMP scale up. We made kilograms quantities of the non-GMP scale up, so we have that capability. we can do it very quickly to make those, scale up those type of compounds. In addition, we are supporting a number of companies performing clinical trials for PROTACs. So our development and manufacturing colleagues, they have the capability to manufacture GMP batches of PROTAC compounds. One of the customer, we supported them. We delivered just in, very quickly some 650 kilograms of GMP batch compounds to support clinical trial studies. So we have this end-to-end, if you will, this one-stop solution from discovery to development to manufacturing for PROTAC type of compounds.
Okay, very good. We certainly have time for more questions. How can we improve target selectivity for covalent drugs?
Okay, so yeah, the covalent drugs, I mentioned that prior, before, people reluctant to develop covalent drugs because the perceived toxicity. The perceived toxicity often from the off-target effect, because this reactivity of the covalent, warhead, they can often react with a number of other proteins. So how to improve this target selectivity? So number one, people, customers are using more structure-based drug design. There are more crystal structures available. There are better tools to design your compound to have better binding for the target protein or the mutant protein than other proteins. So that's number one, using computer-aided drug design to design better molecules and better position, your warhead. That's number one. And also there, for the PK profiles for covalent drugs, they are quite unique. They're not following the same as reversible inhibitors. So often what you do is number one, you design more potent binders to the target protein. Number two is you fine-tune your warhead, and the warhead is not the most reactive, is the best. And so you need this right balance. It's reactive enough, but it's not too reactive, for the target protein functional group. And that needs to be tested in the PK/PD studies. And so what you want to have is the very quick on target covalent bond forming and a slow off target reaction with, or if the bond forming with the off targets. In addition, for reversible drugs, we often want very stable plasma stability and so on. But for covalent drugs, you need a short window. And oftentimes, the half-life, you don't need that long. You just want to, as quickly as possible, react with your target protein. And then once it's done, you want the compound as quickly as possible, metabolized. So it will not have a chance to have non-selective off-target reaction. So that's why I say you also have to make the compound, test in vivo to find out. So there are a lot of tricks to improve the selectivity for your target protein.
Okay, so that's covalent binding. Let's turn to another topic, nucleic acid delivery. How do you deliver nucleic acid therapeutics to target organs other than the liver? Yeah, so that's yeah. So it's right now, as I mentioned, the currently approved 5 siRNAs, they're all targeting liver, and so the liver right now is the only successful for the siRNA, but in clinical trials there are other siRNA targeting non-liver, extra hepatic tissues, and for the ASOs,
there are ASOs targeting other organs, including the CNS, through intrathecal injection. So it is possible, but how do you have a better tool to do this? So there are research papers or clinical trials. They use one is use transporters, and some use large molecule protein transporters help transport oligo nucleic acid therapeutics into the into the CNS, and also they're using their research work carried out using small molecules like certain fatty acids or certain steroid type of delivery to help the compound deliver to CNS or other non-liver organ tissues. So there are tricks, either through the different administration route or targeting conjugation through large molecule peptide or small molecule conjugation to help deliver your compounds to different organs.
Yes. Just a couple of questions then before we close. Dr. Guo, you presented an exciting and impressive range of therapeutic strategies. I'm curious whether those include any activity at WuXi in the area of CRISPR and base editing or precision DNA editing. As a chemist, as a former chemist, I'm sure you're really excited by the ability to perform these chemistries on the genome, but I'm not sure if that has any active any activity as far as your company goes. Maybe you have to draw the line somewhere. What do you think?
Yeah, thanks, Kelvin. First of all, we are a CRDMO, CTDMO company. So we use our platform to support customers' research and the development, manufacturing. We don't do our own, drug discovery. So that's the line we draw. And for supporting customer research, we do add new capabilities to support customers' research. And you mentioned CRISPR gene editing technology. We don't do that as a drug discovery project, but we have that technology to develop what we use as animal models. We develop new animal models to put in the make the gene editing in the animal models. So yes, you have the animal model to support customers drug discovery projects. And also for the gene editing, CRISPR, we don't do the whole package, but we work on, for example, we synthesize the sgRNA, to help customers to package with their CRISPR package for their work. And we also make the lipid nanoparticles, because similar to ASO siRNA, and we have made some several thousand lipid nanoparticles for customers. Lipids, they can make the package to deliver their gene editing package.
Very good. Final question from me, hopefully a positive one. Just before we went on air, you told me that WuXi had grown from maybe 4,000 employees when you started at the company to now some 40,000 global employees. Can you just talk about the growth of the company? Where have the real growth points been over the past decade?
Yeah, thanks, Kevin. Yeah, so WuXi started in year 2000, and so we are relatively young, 23-year-old, and so the company started with 4 co-founders, our CEO and 4 a total 4 co-founders, grew from there to now to some over 40,000 employees globally, we have global presence, and the growth came from customers' needs, and so we have our platform to cover from end to end, from research to development to manufacturing. So this way, so it's a one-stop end-to-end, service, and also, we have the platform to help customers to speed up the discovery and also shorten the overall cycle time for the development and the manufacturing. So that's mainly from the customer need in the drug discovery, and so that is the that has driven our growth, and we use our platform to support the customers' drug discovery needs.
Very good. That's a great note to end on, and I believe that's all the questions that we've received. Yes, indeed. So that does indeed wrap up today's webinar. It will be available, as I mentioned, on demand in the next couple of days. So you'll be able to watch this back again, study the slides in detail if necessary, and of course share the links, please, with your colleagues who might have been enjoying a summer's day or unable to attend as they had wished. So we hope you will communicate that to your colleagues. Huge thanks to Dr. Tao Guo. Really good to meet you. Great presentation, and thank you for answering all those questions. And of course, our sponsor, WuXi AppTec, a very successful company, will be working on at least one more webinar towards the end of this year with them. So look out for more news on that. Thanks to the Gen Multimedia team for hosting, led by Liana Jabs and David Mosley today, and our friends at WorkCast as well for providing the platform for you to enjoy the webinar. But most of all, thanks for you for tuning in, and hopefully you enjoyed and found today's presentation informative. So all that remains for me to do is to say thank you for everybody at Gen. I'm Kevin Davis. Thanks for watching. Goodbye for now.